Cool. So moving on. So what I'm kind of interested in is like not only the bacteriophages, but also bacteria they are interacting with. And it has been kind of well established that bacterial communities, the microbiomes, are quite important there, important for the plant health and growth. And if you think about it, you have lots of examples, symbiotic plant growth promoting bacteria like nitrogen fixing rhizobia, but you also have pathogenic bacteria that can be seen as sort of invaders of bacteria. What I mean with the invaders is that they don't necessarily get to the plant right away. They have to actually go through this microbiome to be able to infect the plant. And this microbiome sort of forms the first line of defense. And then there might be multiple different ecological mechanisms that are kind of preventing these invasions. So there might be resource competition with the native microbiota and the competitor. They might be using similar kind of ecological niches, which might prevent the invasions. They also, bacteria produce all sort of toxic compounds, antimicrobials, and this competition might prevent invaders to establish themselves. You also have lots of trophic interactions, not only bacteria, but for example, protists, bacteria, nematodes, all feeding on bacteria. And this talk, I'm going to be focusing on the role of the phages in this sort of like invasion resistance, or kind of like shaping natural microbiomes and pathogen invasions. And my kind of model bacterium is called Ralstonia solanacearum. It's kind of causative agent of bacterial wilt disease on multiple, I mean like hundreds of plants, but multiple agriculturally important crops like potato, tomato, banana, aubergine, and so on. And it's sort of like generalist bacterium. It lives in the soil, and I'm quite happy to be there in other aquatic environments as well. And how it kind of gets to how causes the infection is kind of gets through the microbiome, gets and interacts with the plant. There's lots of ways it produces effector proteins to evade plant host immunity, gets inside and start proliferating. And basically it just blocks the water circulation. So it's very slimy bacteria that you can see lots of exopolysaccharides produced, and it's kind of tripping on a tilted plate. And we've been working on sort of collection of phages and bacteria in UK and also in China. And with this talk, I'm going to focus on the China part of we've been working there recently. And I have like three sort of general questions. So I'm kind of interested in like ecological dynamics between my pathogenic bacteria Ralstonia and phages, and specifically if the phages could be kind of controlling the pathogen naturally by just infecting and controlling the population densities. Then we'd be kind of moving on, trying to kind of think about the whole sort of virome effects, not only thinking the pairwise interaction between the pathogen and its phages, but also everything else that is surrounding, other bacteria that are actually interacting with the pathogen and the phages of these other bacteria, and how these sort of more complex ecological interactions might be affecting if we get disease or not. And then I'm sort of evolutionary biologist by trade, so we're interested in phage bacteria coevolution, and maybe if this evolutionary changes or the selection is changing the ecological interactions between bacteria and phage. And I'm going to first start with the sort of ecological questions of density, top-down density control, and this is work by Chinese PhD student coming out from Nanjing Agricultural University. And then this is sort of stemming from one big experiment they did a few years ago, sort of like rhizopod experiment. And the idea here was like, we don't necessarily know which of the plants on the field get infected or not. We only know that once the infection has set in. And then we basically just did field study where we sowed tomato plants on field, and we used this sort of rhizopod system that has sort of inner layer that had nylon bags filled with sand. But you could sample these nylon bags, and then they were kind of interacting with the plant roots, and then they were considered as a sort of subsamples of rhizosphere. But that kind of allowed us to take samples every week of all the plants. And after week 6, we just chose subset of healthy and diseased plants from our collections. And then we went back in time and we chose all those samples we collected and sequenced them and looked what's in there. And the kind of main question being, is the sort of microbiome development very different between healthy and diseased plants, and kind of focusing on the effects of the phages, bacteria, and looking at the pathogen densities as well. And this is just looking population density data between these two sample cohorts. And here on the left, it's qPCR data on Ralstonia, and we see quite a nice increase in bacterial density around week 5, and this is associated with the disease plants. Interestingly, when we look VO2 abundance of Ralstonia phages, we generally have higher Ralstonia-specific phage densities in healthy plants. So it suggests that for some reason, it's kind of weird that it happens already at the time point 0. So that's sort of like luck effect in a sense. We just by choosing randomly 4 plants, they had higher abundance of our pathogen-specific phages in those soils. And then you can look this as a sort of phage bacteria ratio as well. And it kind of looks, and I'm using the metagenomic counts data, and it shows that overall you have more phages that are able to target this pathogen in healthy plants. So I kind of suggest that there's some interesting effect there. And then if you look some individual phage, Ralstonia phages, we can find here, there are few of them, like one of them, RSL1, which is Champomyovirus phage, that has been shown to actually have this sort of biocontrol effect. You can produce that in the lab, and it's quite effective at killing the bacteria in the soil and protecting the plant. So of course, this is sort of correlative data. We didn't do an experiment. We just took healthy and diseased plants and tried to kind of observe if we can find any differences there. So we've done more work on just like more controlled experiments where we add in the phage or we keep them have control plants. And this is done by Shifeng Wang, another PhD student, former PhD student who is now leading her own research group in Nanjing. And he did sort of like experiment, what kind of applied question in mind as well. Like what happens if we add in the phages sequentially? We don't only add them, in the beginning of the experiment, but we keep adding those into the soil. Can we get better density control of our pathogen compared to if we apply them only once? If we apply them once or around day 9, or then 3 times where we added them day 2, 9, and 16. We use this sort of 4-phage cocktail we can develop earlier. And what we find is that we get reduction in disease incidence with the kind of number of phages, the number of times we apply the phages. There's not a big difference between if we apply 2 or 3 times, but there's higher reduction in disease incidence, disease index, if you apply the phages 2 times. And then interestingly, we can see the same when we look at pathogen and phage population density dynamics. So when you don't have any phages, you have quite high pathogen densities, and they're kind of reduced more with the number of phages or the number of times you apply the phages. Similarly, you can find more phages when you apply them more sequentially and more often. So it kind of suggests that the kind of strength of the phage pressure we apply is having negative effect, more negative effect with the bacterial growth. So there seems to be this sort of top-down density control we can use in a biocontrol of this pathogen. So kind of like interesting, like this is kind of cool. We're just looking at these few species that are interacting in this complex microbiome. We can see some effects, but then of course there's other bacteria there. Some of the bacteria are antagonistic to our pathogen. Some of them are actually facilitating the pathogen growth. And of course, these bacteria also have phages. So we've been kind of looking what happens around the microbiome. The same experiment, just took the 25-day sample point, and this is just showing the diversity of the bacterial communities. What we kind of see is that there's increase in the diversity with the frequency of phage administration. And we can also go down and look what are the kind of changes in phyla abundances. What we kind of nicely see is that on black you see Ralstonia 16S sRNA sequences, and you can see that they're actually going down quite nicely with the frequency of administration. And interestingly, you see also changes in the proportion of other bacterial taxa as well. So when you're kind of like taking out this Ralstonia, you can properly like vacating niche space. So other bacteria can come up, grow up, and then take up the space. And you kind of get this sort of community-wise change. And interestingly, there's few groups we've been kind of focusing. One is actinobacteria, which is known to produce lots of different antimicrobials. And then we use lots of antimicrobials in our medical world, which are produced by actinobacteria. We actually see increase in the relative abundance of actinobacteria. And then basically the pathogen density is negatively correlated with the Streptomyces abundances. We also done experiments where we take these Streptomyces species and we test directly in the lab if they are able to suppress the pathogen. And then yes, they are. They're quite often very antimicrobial. So it kind of suggests that applying these phages, you kind of changes the whole makeup of the microbiome, and not only the taxonomic composition, but also the functioning of the microbiome. So maybe kind of going back to the Rhizobox experiment as well, and I kind of see like, do we see anything similar there? This is just looking at the bacterial community composition between healthy and diseased plants. We also kind of able to pick up some antibiosis production genes. It seems to be in higher relative abundances in healthy plants. And if you look at the kind of sort of whole virome, we do see kind of clear change in here as well. So initially, the whole virome is very different between the healthy and diseased plants. And interestingly, this difference is kept throughout the experiment. So essentially, the viromes of the healthy and diseased plants are very different, and you have different proportion of functional genes there as well. So the kind of idea we want to test is like, maybe there's some sort of secondary phage effects that are affecting indirectly to the infection. So you might have bacteria that have antagonistic effect on the pathogen, but then those bacteria also have phages. And then these phages can actually then indirectly reduce the antibiotic effects by these pathogen suppressing bacteria. So we tried to kind of dig out this sort of interaction from the metagenomics data, just looking species abundance correlations in time. Again, looking that there's kind of different nodes that are sort of different functional groups. You have Ralstonia, the pathogen in the middle, you have Ralstonia specific phages, and the sort of colors in here are denoting different sort of negative or positive correlation. And we have bacteria that are basically inhibiting Ralstonia. Some bacteria are having a positive sort of correlation. And of course, all these bacteria can be associated with some of the phages, predicted phages in here as well. And of course, this is only correlations you can do and try to put all this together and then use structural equation models and try to see like what are the kind of relative strengths of these different interactions in your community. And what you kind of find out is that with healthy plants, you have quite strong negative correlation on Ralstonia pathogen densities by Ralstonia specific phages, but also pathogen suppressive phages. So you have sort of like dual effects in these healthy plants. You have lots of phages that are able to kill the pathogen, and also lots of bacteria that are sort of potentially antimicrobial producing bacteria. Compared to in the disease plants, you don't have the same correlations. The strongest relationship you get is that you have lots of phages that can actually infect these pathogen suppressing bacteria. So it kind of suggests that maybe these phages are able to kind of nullify the effect of pathogen suppressing bacteria in these communities in the disease plants. So this is all good. It's just like metagenomics data, spurious correlations. Can we actually test it directly in a lab? And we kind of went back to the same field, try to isolate bacteria that are antagonistic to our Ralstonia and the phages for those bacteria as well. And we were able to pick up three bacterial species and 3 phages for them. And we want to test if you can actually reproduce this effect we observe in the disease plants. What we find is that in the lab, we do see this effect. So essentially, this is just the Ralstonia growth as a like RFP signal. And let's put the magic thing. Here we go. So basically, Ralstonia abundance is if you add just the phages that are basically specific to this other bacteria, you don't see any negative effect. These bacteria alone has quite strong negative effect on Ralstonia, so they're all reducing Ralstonia abundances a lot. But when you add in the phage that are able to infect these pathogen suppressing bacteria, this effect is less strong. So suggesting that these phages are killing the inhibitor bacteria and kind of like making the inhibition weaker. You can then do the similar experiments in greenhouse as well. So this kind of with sterile substrate, just kind of simplify things and just to see effects. We see similar effect in disease incidence. So basically, this is just showing the effect of suppressing bacteria. So they're really protecting the plants, but when you add in the phage there, you can lose that effect. Similarly, you can get a similar kind of pattern with the pathogen densities as well with the qPCR. So it really suggests that the phages can help. The wider virome is important for these infections. Kind of lastly, I'm going to just show quickly, like we do kind of interested in mechanisms as well. What is the mechanism of resistance? What are they using for infections and so on? We're interested in like, what if these bacteria are evolving also? We do these experiments in the plants at the time scale of weeks. That's enough time for the bacteria to evolve. There might be selection happening as well. So it's not only necessarily ecological effect. We've been doing this sort of greenhouse experiments with the same 4 phage cocktail, but using them independently. What we see is a clear increase in resistance, not as clear with all the phages, but compared to ancestral phage and control treatments, they become more resistant, better at growing. And interestingly, you start to see these sort of life history trait correlations. So actually, when they become more resistant to the phages, they kind of have sacrificed their virulence. So they become less able to infect the plants. So in here, the pathogen virulence is actually measured as ability to infect the plants. You also see trade-off with competitiveness. So this is just looking what sort of resource use range these evolved bacteria have. And actually, they cannot utilize that many carbons anymore after a while. And then they become less competitive in that sense. You can do pair-based competitions, direct competitions with fluorescent strains as well, and you see similar patterns. And of course, we've done some work on trying to figure out like how do they evolve resistance. And then sometimes you get these nice sort of phenotypic effects. In this case, we get sort of phages are sort of enforcing selection on these sort of non-mucoid small phenotypes, which is seen with many of the bacteria. Interesting thing is that these guys, they lose their virulence completely. So even if you take these bacteria, you put them in the needle and you inject them in the plants, nothing happens. They cannot infect the plant at all. And you can look into sort of like what sort of mutations you can identify. For example, in one case, we find pilus mutations. This is actually the original mucoid type that has pilus mutation. So again, like probably suggests that phages are using the pilus as a receptor. We haven't verified this by engineering mutants yet, but it can suggest that might be the case. And you also see phenotypically reduction in twitching motility, which might affect negatively on plant colonization. You also see mutations in sort of like chromosensin gene, which is sort of blocking the master virulence regulation with this bacteria. So it's interestingly, it kind of suggests that there's another way to kind of become resistant. So this is just like block the whole virulence gene expression. And these are the small colony types. And even if you inject these to the plants, they don't kill the host. And the reason is probably because they don't activate the quorum sensing regulated virulence mechanisms at all inside the plant. So coming to the end, kind of my sort of take home message, like it's kind of preaching to the choir to say that phages matter in here. Like I was in an ISME meeting and I want to make point that most of the ISME meeting stuff is about diversity and equality, and we can see species frequencies and abundance changes. But at the same time, we have changes in genotype frequencies. Species are evolving. They are evolving in a matter of few weeks, even in the soil. So actually what we probably see is a kind of mix of ecological and evolutionary dynamics. So you have changes in species abundances and frequencies, but within each species, you have also changes in different variants, mutations, and then mobile genetic elements are moving and changing the makeup of the populations. And then maybe I was kind of making the point in the ISME meeting that phages are important. Maybe in here, this meeting, I can say like bacteria are also important. So not only the phages, but how they interact with the bacterial community actually matters when we kind of go into kind of trying to understand these sort of microbiome level things. And this is the team who've been doing all the work, and thank you for your attention.
Thank you for the talk. I'm Lotta-Reena from Jyväskylä, Finland. I was just wondering from the perspective of phage addition, and also from the perspective that phages are there, and why do they not control for the bacteria populations to go non-virulent? That when you add the phage, how long do they stay in the system, and how widely do they spread in the plant?
Yeah, so second question, we haven't been able to actually sample how they are spatially distributed in the rhizosphere. But like when you do the experiments in the greenhouse, you're watering the plants quite a bit. The soil humidity is like 70%. That's really good for tomato. So they can spread, but of course they're inactive, they don't move. But we don't know. Phages can survive quite long times. Most of these phages come from the soils and then rivers, so they kind of adapted that sort of harsh environmental conditions. So like with the phage addition experiment that lasted 25 days, and you could easily recover phages after the 25 days after one initial application.
Hello, thank you for the nice presentation. I am Sultana from University of Manchester. You mentioned that phage resistance is associated with host gene regulators. Have you been tested the relationship between phage resistance and these gene expression regulators?
So we haven't looked, well, we have done some transcriptomics experiments just mainly to kind of look, I have a few slides in here, just like looking at some of the defense systems. So we kind of looked at defense systems, but we know that lots of the bacterial genes are actually activated or downregulated when they are faced with these phages. So this is the same phage in bacteria. What happens after six hours of the infection? And we get like upregulation of abortive infection systems, restriction modification systems. When you repeatedly expose this bacteria to these phages, you get these mutations that are actually giving them genetic resistance. But you get changes in like lots of the motility and virulence related genes are actually going down on the bacterial side, and we don't know like what's going on. There's quite a lot of things happening when you just expose them to the phage even for a long time, and not all these genes are linked to the defense systems or resistance.
Oh, okay.
But yeah, it's a good question, but yeah.
Yeah, I mean also, have you like found any gene like responsible for gene expression regulator, any specific gene, or no?
We haven't looked at that detail yet, but
okay.
that's really good. But they do have like lots of, they have CRISPR-Cas, and not all the strains, but lots of around 10 to 12 defense systems per strain in the Chinese fields.
Thank you.
You know, that way you said that one of the problems is the bacteria that are actively suppressing pathogens and then being killed by phages. How is that happening? Where are those phages coming in from, or how is that dysbiosis being caused?
Yeah, so I think like the Ralstonia can survive in the soil quite well, and then probably like lots of the Pseudomonas bacillus spore-forming bacteria, especially, they can survive in the soil. So like Ralstonia probably quite often is human introduced. So along with the crops, or like with seed potatoes, you can transfer it between different fields. But they can survive in the soil quite a long time. So fields that get infected in China by Ralstonia, you normally just change the crop from tomato to strawberry or something else that cannot be infected. But then you can still go like 10 years after and you can isolate the Ralstonia there. So it's very well adapted to the environmental conditions as well. And then that's sort of like hard to eradicate in that sense. But yeah, I mean, like we haven't looked yearly variation on how much the microbiomes are changing. This is sort of like only like 6 weeks. And then it might be that there's big differences actually at that scale as well with the years.
Okay, well, I think in the interest of time, we should probably move on, but thank you very much. That was a very fascinating and interesting talk.